sure people keep joining, but I want you to have your time, and then hopefully we have some time for questions at the end. But I'm super excited today to have Dr. Caleb Peterson present here. He's an assistant professor of research in neurology with the ANSYS Bioimaging Lab at WashU in St. Louis, where he's been studying the impact of biological aging on brain health and cognition in people with HIV. He's received his PhD in chemical and physical biology from Vanderbilt, where he specifically specialized in structural and functional MRI, particularly in neurodegenerative movement disorders, including Parkinson's. He's been awarded at this point an F32 fellowship. He now has a KO1 Career Development Award from NIMH, and he also received a fellowship in biomedical research from AMFAR. He's a member of the STAR training program run by UCSD. In addition to working with brain imaging, his current projects have now integrated epigenetic changes in DNA that occur with aging and disease. I'm super excited to hear what he's been up to. So I'm going to hand it over to you, Kaylin.
Thank you so much, Leah. I appreciate the introduction, and I'm happy to see so many friendly faces here. Let me share my screen. Okay, hopefully everyone can see that. Looking good? All right. So my talk today, I gave it kind of a wordy title because it's covering basically the broad areas that I've been working on as a part of the ANSYS lab for the past 5 years. But the core areas are in aging, neuroimaging, and cognition in people with HIV, especially people who are getting older living with HIV. And I'll tell you just briefly, Leah already covered a lot of this, just about where I've come from. So I did my PhD at Vandy, and I did a short postdoc for additional training in radiology at Vanderbilt Medical Center. And then I joined Bo's lab in 2020 and have been doing a postdoc since then up until this year when I started on my K project, or last year rather. And so this little word cloud thing gives kind of a cluster of the areas that I've been working on, both for the postdoc projects and now for the ongoing areas of research, which include epigenetics and also continued studies in neuroimaging. So in part one, I'm going to give the imaging portion of this talk and discuss how we can measure biological aging in the brain using MRI. And then part two, I'm going to transition to these more recent projects where I've taken on a new area of training. I'm learning how to study DNA methylation data and how to use epigenetics to measure biological aging. So I know that there are some people on this call who are experts in this area. So because this is pretty new to me, I would absolutely love to get people's feedback and advice. I saw, among others, I saw Carrie Johnston on here and other people who are working in the DNA methylation and clocks field. So feedback on this unpublished data is very welcome and appreciated. Okay, so part one, I'm going to start by giving a little bit of background that you probably all know on HIV. So the number of AIDS deaths has declined dramatically in the past 2 decades, and so have the number of new infections. And the good news is that globally, over 3 in 4 people have access to antiretroviral therapy, which means that despite the reduced deaths and the reduced number of new infections, the number of people worldwide living with HIV continues to grow because people are living longer and life expectancies are now typical for people with HIV. I'm giving you the color codes that I'm going to use here for this first half of the talk. So I'm going to use green for people without HIV or controls, and yellow for people with HIV who have undetectable viral load, and then red for people with detectable viral load who are a minority in the US and in our studies. So this means that HIV is now a disease that is complex in the way that it presents as people get older. So a majority of people in developed countries who have HIV, as you know, are now over 50 years old. And this highly publicized fact means that we now have a new population who are aging in a way that we've never seen before. And this epidemic in the US tends to be concentrated in more marginalized communities, economically or socially marginalized. And all of this combines with a highly prevalent set of comorbidities to produce a lot of different potential drivers of changes to brain health. Everything from substance use to heart disease to co-infections to the direct effects of HIV itself. And of course, we know that HIV can affect cognitive function, can affect brain health. It probably gets into the brain on a Trojan horse mechanism in immune cells and monocytes, and then sets up shop as a latent infection, creating a viral reservoir where it can lead to persistent inflammation in the central nervous system. And this can lead to the establishment of those neurocognitive disorders. Of course, dementia due to HIV is increasingly rare with ART, but more mild forms of cognitive impairment remain relatively prevalent, affecting maybe 1 in 3 people with HIV, although estimates vary. So the overarching question of our research here has been whether this cognitive impairment is related to accelerated brain aging. And I'll try to define what I mean by that more specifically. So the data that we're working with has been acquired at this site at Washington University in St. Louis over the last 16 years or so. So we have a large cohort of individuals with neuroimaging and also with neuropsychological profiling, clinical profiling, blood draws, and other resources that we can utilize to better understand the aging process in people with HIV. So as you can see, our cohort includes over 200 controls, 200 undetectable people with HIV, and about 100 detectable viral load people with HIV. And then for a lot of those, there's also longitudinal follow-up data available where people have come in for additional scan time points and study visits. So it's a pretty large and well-characterized data set. And you can also see that there are significant demographic differences in these groups. For instance, we have differences in baseline age, in the percent male and female participants by group, where we have more male participants among our HIV set, and differences in race, where we have more African Americans represented in our HIV group as well. Differences in baseline levels of education, although in this set that was not significant. And then, of course, we look at our HIV clinical metrics, and we can see that our detectable viral load group, of course, has lower CD4 T cells, and there are differences in the CD4-CD8 ratio. So going forward, I control for the demographic, core demographics, so age, self-identified racial group, and sex in all the statistical analyses. So we have a really powerful imaging toolkit to understand what's going on in the brain, and some of these tools include structural MRI, which is high-resolution three-dimensional scan of the brain that allow a gray matter and white matter contrast to define where the boundaries of the cortex are and the subcortical structures. And that lets us get brain volumes region by region and morphology values like cortical thickness. And then we have diffusion MRI, which is a specialized technique for measuring the health of white matter by looking at the diffusion of water molecules. I'll talk about that later. And then arterial spin labeling, or ASL, is an MRI technique that can non-invasively, in other words, without a tracer, measure the flow of blood into the brain, which is another biomarker of brain health that changes with age. Okay, so I want to go through some of the results from the previous studies that we've done looking at these three MRI measures and talk about what those have to do with HIV and viral load. So in the past, a number of studies have identified differences between the brain structures of people with HIV and people without of a similar age. This includes both the cortex on the surface of the brain and subcortical structures. But the biggest consistent theme has been variability. Different studies have found wildly different brain regions, wildly different patterns in time in what areas of the brain are affected. Um, and it's been hypothesized that these patterns of cortical change might resemble just an acceleration of typical aging process, or maybe the early onset of age-associated neurological diseases. And our thought in approaching these studies with that literature in mind was that maybe some approaches from data science and machine learning could help simplify the problem. So you could say our overarching hypothesis was that machine learning using whole brain MRI will identify differences in aging rates associated with HIV infection and detectable viral load, and also with cognition. So fortunately, people have done really clever things with training machine learning models to estimate a person's biological age from their brain age. So the general concept of brain predicted age is that you train a normative model on a large neuroimaging data set representing people all across the lifespan. And you cross-validate that to make sure that it can accurately predict age. And then you test it on holdout data. And the idea is if you show the model brain scans of people who have neurological disease, in general, it will err on the side of older appearing ages. In other words, the gap between a person's predicted age and their chronological age can be taken itself as a biomarker of brain health. This is the concept of the brain age gap, or BAG. There are other names you might see for it in the literature, like brain PAD. It all means the same thing. And if the BAG is positive, that generally indicates pathology. And if it's negative, that might indicate resilience. And this just helps simplify and focus all this complexity in the neuroimaging data sets into an outcome measure that's easy to interpret. The model that we chose, and there are quite a few available, each with pros and cons, but the model that we usually work with is called Deep Brain Net, which is a convolutional neural net that was trained on a huge number of participants across lots of different sites and studies. And the advantage of Deep Brain Net is it takes minimally processed structural brain scans as input. All that is required is that non-brain tissue be removed and that the brain be aligned to a standard template. And so this eliminates a lot of the differences in pre-processing that might otherwise influence the outcome of the model. So once again, the output of this model is what we call the brain age gap, which is the difference between chronological and biological age, where positive is thought of as bad. Okay, so on our data set, this produced a pretty accurate estimate of age in controls with an average error of about 5.2 years. The connected points on this plot are those individuals with more than one scan. And as you can see in our controls, chronological age on the x-axis is faithfully mirrored by predicted age on the y-axis. And we were interested to see that people with undetectable HIV still in general showed a significant acceleration of brain aging, especially when you looked at individuals older than 50 years old. And part of this may be entirely a legacy effect of early infection, where people may have taken a hit to overall brain health and then stabilized over time, or it could have something to do with ongoing processes. We saw that people with detectable viral load looked a little bit older, although this was not significantly different between the detectable and undetectable groups, which sort of supports the idea that this might be a legacy effect. And then I want to just illustrate that the variability with any one of these groups is very high. So there are obviously people who are aging and aging very well and resiliently, and there are people who are maybe having more impact on their brain and on potentially on their cognitive processes as well. So big within group variability. However, these between group differences remain significant. We did see that this is associated. Oh, I see Ron has asked a question in the chat. I think I answered that, but some points are connected by lines because those were longitudinal. Yep. Oh, and Leah has a question in the chat as well, which I'll address real quick. For brain age gap, the measure is derived. I guess this isn't a question, Leah.
But it is, sorry, I just want to understand why it's actual, like it's an actual minus a predicted, but typically it's always divided by some measure of variability. And that's very similar to when we think about normative based regression. So I just didn't know why it's just the difference and nothing that
Ah,
accounts for variability. Sorry if I wasn't clear.
yeah, that's a good question. I think you could z-normalize it if you wanted to, and we have done that when we wanted to compare across different models. We've z-scored the brain age gap or the epigenetic clocks that I'll talk about later. I think the reason that it's just taken as a simple numerical difference is just that it provides kind of an easy to interpret value that if the goal was to make this into a useful individual biomarker, it would be very helpful to say so and so has a brain age gap that's plus 7 years or minus 5 years that gives people an intuitive sense of what those values mean. But I understand your point about normalizing to the within group variability. That makes a lot of sense. Does that answer your question?
Yeah, I was just curious if that helped or didn't help. And I know it puts it into a different metric, but it would still keep it on some sort of Z or whatever scale to have it in the same thing. But if these are being done and normed to external databases, then that would also have variability. And I just didn't know if it would be more sensitive or not, or maybe it
Yeah.
doesn't matter.
Yeah, that's really a good point. We haven't normed these to any sort of external data set. We've just when we have done normalization, it's been to the within group variability. Yep, yep. Okay, so just really quick, we do see that this brain age gap corresponds with some of our key cognitive measures. In the case of the bag that we get here, we saw that it was particularly associated with psychomotor slowing, but also with executive function, the cognitive composites that we get from our neuropsychological battery. So it seems to have an association that is meaningful in the cognitive realm. Okay, so that can tell us a lot about the structure of the brain at the gross anatomical level, but MRI can also measure differences in the microstructure of the brain, particularly in white matter, because in white matter, where axons are in large parallel bundles, and these bundles are encased in myelin sheaths, the movement of water molecules is very constrained by the microanatomy. And diffusion MRI allows the measurement of constrained or unconstrained water movement in such a way that we can measure the overall health of a white matter tract. And this is a measurement that takes place at the micro scale instead of the visible anatomical scale. We know that HIV is associated with damage to white matter, including demyelination in some cases, or the loss of white matter volume. And we hypothesized that if you looked at brain aging based on white matter, it would be accelerated with HIV and even more accelerated with detectable viral load. So in this case, there wasn't an available model, so we trained one using a publicly available data set from Cambridge, the CAMCAN neuroimaging set. And we gave the model data on water diffusivity and anisotropy, and we trained a Gaussian process regression model. And then we tested it on a population of people with HIV and controls. And then that allowed us to predict the brain age gap from the white matter. And then we compared the results of that model output with our existing well-validated deep brain net model to see if results were similar. And then finally, we tested whether white matter aging was accelerated, whether it was linked to cognition and to other potential predictors. So briefly, when comparing to the well-established model, we saw a strong significant relationship between the two. So our new model is on the x-axis and the existing model is on the y-axis. And as you can see, the people with HIV with detectable viral load showed up with a higher average brain age gap. And to quantify that here, let me plot the three groups again with real age on the x-axis and predicted age on the y-axis. And hopefully you can see that the detectable group in red has a steeper slope. This is a more modest effect size, but it's also significant. So it adds up to about 1 year of quote unquote brain age for every decade of real age. And you can see this here in the violin plot. We also found that it was associated with cognitive function, in particular, in people with detectable viral load. There was a group-specific interaction where they had worse overall cognitive function with greater BAG. So I think it would be interesting to follow up on this to ask whether BAG predicts longitudinal cognitive trajectories, whether people will be stable, decline, or improve. So finally, neuroimaging allows us to measure this dynamic process of blood flow in the brain as either a functional measure or an overall measure of brain health. And in this case, we're using a method called arterial spin labeling. Jules, yeah, we did look at nadir CD4 and current CD4, and in this study, we didn't see either one of those associated with the brain age gap. Thank you. So the cerebral blood flow is measured in a clever way by using radio frequency pulses to magnetically label blood water in the neck, which then flows up into the brain. And an image of the whole cortex of all the gray matter and subcortical gray and white matter is taken. And this is compared to an unlabeled image. And when you subtract the two of them, the difference of magnetization is proportional to the blood flow. And then you can plug that into a numerical model to get an estimate of cerebral blood flow. And we already know that cerebral blood flow declines with age. So the question was, will CBF decline at a faster rate across the lifespan in people with HIV if they have detectable viral load? So here are our controls, and you can see the expected linear reduction with age in mean cortical CBF. And the blood flow for the people with undetectable HIV, we're happy to say, was exactly the same. However, the mean slope of people with detectable viral load, as we look at older and older ages, was significantly reduced in an interaction effect. So it seems that the combination of detectable virus and older age is particularly troublesome for cerebral blood flow, which you can see in the bottom right figure of people over 50 who have detectable viral load. The cooler colors mean lower blood flow, and you can see that the patches of hotter colors are reduced compared to the younger individuals and compared to people with viral suppression. Just to cover this very briefly, this was not a regionally specific effect. We looked in every lobe of the cerebral cortex and in some of the deep ganglia, and we did not see really any difference in this overall pattern where it was the people with detectable viral load who had maximum blood flow loss. So I've made a big deal about HIV and detectable viral load, but as I alluded to at the start of the talk, this is more complex than just the direct effects of the virus, because we also have at work a lot of different factors, including comorbidities and including social factors that could be influencing brain health. And I want to call attention to just a couple of these. I know that we don't have time to talk about everything on this chart, but I want to really quickly look at the impacts of cardiovascular health, hepatitis C co-infection, and some of these social determinants of health. So with regards to cardiovascular disease, we looked at the Framingham score, which is a 10-year prediction model for major cardiovascular events. And we saw that the greater the Framingham score at the study visit, the greater the brain age gap in people with HIV, suggesting that the risk for future cardiovascular events is also associated with present brain health measures. Let me go back one because I see more questions in the chat. Cerebellum white matter. So someone asked whether cerebellum white matter versus CBF was measured. We didn't look at that. Usually we only assess blood flow in the gray matter because it's higher and a little bit more accurate. But I can't remember if we looked at cerebellar gray matter. There's a question about vasomotion. I am not sure. I don't know whether there's any data to support changes in. Actually, I don't even know what vasomotion is, to be honest. So if someone wants to explain that to me, you're welcome to. And the use of statins, we did not look at statins as an exposure, but I think that the use of statins is incorporated into the Framingham risk score model. Thanks, Scott. So it's contraction of small blood vessels. And then Jules also mentioned semaglutide. We have not looked at a GLP-1 or Wegovy or what have you, but I know that there are groups out there who are doing clinical trials, and I'll talk just a bit about that in a later slide. Okay, moving on. Hepatitis C co-infection is another common comorbidity of HIV, and it's very prevalent, affecting maybe 1 in 5 people with HIV in the US. And we noted that there was actually quite a significant brain age gap difference between people who are hepatitis C positive and also HIV positive, and those who only have HIV. But that's good news because hepatitis C is a treatable risk factor. Lastly, I touched on the social determinants of health. We looked at whether education was associated with differences in brain age gap, and we actually found a protective effect for more years of education in people without HIV, but this effect didn't show up in our HIV positive group. Why that is unclear, but I think that you could argue that the effects of HIV might mitigate some of the aging benefits, some of the resiliency that's conferred by additional education. I will come back to this topic too when I talk about DNA methylation in part 2. Finally, we looked at the Area Deprivation Index, which is an overall measure of socioeconomic status in different US census tracts. So we took data from publicly available databases about participant zip codes, and we measured the ADI in the neighborhood where they live. And we saw this is not actually in an HIV population, but we just wanted to note that there's a very interesting geographical correspondence between higher deprivation levels in red and greater risk of higher brain age gap in the panel on the right. Hopefully you can see how those two map on. And in fact, they map onto boundaries of historical redlining in the city of St. Louis that follow a road called Delmar that cuts through the boundary between historically Black and presently predominantly Black neighborhoods and historically white and more diverse neighborhoods today. So to conclude part 1, we have evidence that HIV and viral load are linked with accelerated brain aging. We think that different MRI modalities reveal different aspects of neurological HIV, and that a lot of these differences are related to treatable comorbid factors. So we think that to give a model of this process, we think that aging with HIV plus other risk factors is probably producing chronic inflammation in the central nervous system, leading to accelerated brain changes and ultimately contributing to cognitive impairment. But thus far in the talk, it's not clear whether these brain changes are reflective of something that's happening at the cellular and molecular level. So that takes us to part two. Let me see if there are questions in the chat. Let's see. Let me look at, have you studied the correlation between the brain age gap and the ApoE4 allele? That's a really good question. We do get ApoE genotype for all of our participants. I haven't looked at whether the deleterious E4 allele is a risk factor for bigger brain age gap in this group, but it would be absolutely valuable to study, or whether E2 allele is protective. The second part of your question was about CSF inflammatory biomarkers. I'll just say, yes, we are looking into inflammatory biomarkers, but that is ongoing, so I don't have any results to report to you yet. Yeah, thanks for the interesting comments, Raha and Jules. Okay, so this brings me to the second part of my talk, which is where have I gone since then? So we've done these neuroimaging studies, but they don't tell us a lot about biology or about mechanism. They're more associative. But is there something going on at the level of the cell that can tell us a lot about what might be occurring with the brain and with aging? So I'm going to introduce the idea of DNA methylation. For anyone who's not familiar, it is an epigenetic modification, which just means a chemical change to DNA that doesn't alter the A's, C's, T's, and G's. Particularly, it's the addition of a little chemical side group onto cytosines that are followed by a guanine. And methylation can regulate the expression of genes if it occurs in the gene itself, or also in regulatory regions upstream or downstream from the gene. It can be changed by environmental factors, unlike the genetic sequence, and by diseases over the lifespan. And this makes it a really useful biomarker for aging. And Steve Horvath and a few other pioneers 10 years ago figured out that you could train clocks, which are models that can estimate a person's biological age from DNA methylation patterns. And they do this in a conceptually similar way. You take a large normative sample, you train it on chronological age, and then you allow the model to make estimates on unseen individuals to see whether the estimates are over, under, or right on the line. So I am just getting into DNA methylation, but there's already a beautiful literature forming around epigenetics and HIV. We know that DNA methylation is altered early on in the acute phases of infection, but also chronically, and that it changes especially at regions in the genome that are associated with aging processes and also with inflammation or immunity. Work from Aaron Sunderman's group, among others, shows the second part. We know that it is altered, usually normalized by antiretroviral therapy, but also by a variety of other classes of drugs that are, for instance, anti-diabetic, anti-hypertensive, or anti-aging. Recent clinical trials from Michael Corley's group have looked at senolytics, including metformin, and anti-diabetes drugs, including GLP-1, which Jules brought up earlier. We also know that it is affected by non-HIV comorbidities and also by social determinants of health in the general population. And finally, it is a marker that changes in both brain tissue and in blood. And because blood is the most accessible tissue, that's where we start. But I think it would be very interesting to look at donated brains and see whether epigenomes are changing. So it is still unclear to what extent methylation is altered in people who have virally suppressed HIV. I think there's some evidence that there is alteration, but it's less than in people who are unsuppressed. Whether it contributes to cognitive disorders, and whether these follow the aging processes that are seen in the brain as well. So these are our first results from this study. These are still unpublished data, so I would love to hear people's thoughts on these. But we have now a fairly decent sample size of individuals for whom we have both MRI and also epigenomes, DNA methylation profiling. So that's 120 plus people with HIV and almost 150 controls. Again, we have differences in age, sex, and race, which are controlled for in statistical analysis. Um, I'm switching to a different color scheme here, so I'm going to represent HIV positive individuals in red and HIV negative individuals in blue, because we're only looking at people with viral suppression now. So instead of 3 groups, it's 2. So just briefly, epigenetic clocks have been around for a while. The first generation was trained directly on chronological age. So these clocks are really repeatable and highly heritable, but they're not necessarily as sensitive to disease states. The second generation of clocks have included more nuanced measures. So phenoage is designed to measure the risk of morbidity factors for various diseases, including inflammatory plasma proteins and smoking. Grim age is actually trained on mortality data. That's why it has that spooky name. And then telomere length can also be estimated from DNA methylation directly. And I put these in red because that's what I'm going to be talking about today, in addition to a third generation clock called Dunedin pace. Dunedin pace is really cool because it is designed to predict the pace of aging, the longitudinal pace of aging from a single time point of DNA methylation. And they did this by taking blood samples from the same cohort of individuals in Dunedin, New Zealand over many decades. So these 4 clocks are the focus of the present study. And so we hypothesized that these clocks would be accelerated in people even with virally suppressed status, that they would correlate with brain health, particularly the brain age gap, that they would also relate to cognitive impairment from our neuropsych battery, and that they'd be modified by social and structural determinants of health. Okay, so I'm just going to give you the top line results from these preliminary studies that we are hoping to publish soon. So number one, as expected, there's a strong correlation between the predicted and actual age. Just to give a point of comparison, for the first generation clocks, that was around a correlation of 0.8. And we get similar, slightly lower numbers for these second generation clocks, which makes sense because they're designed to do a little bit of a different thing. So on the top, you can see real versus predicted grim age and pheno age. You can see some separation between the people with HIV in red and the control group in blue, hopefully. And then Dunedin pace is measured a little bit differently. So here, a rate of 1 means typical aging progression, and anything above that means a faster longitudinal pace. And then finally, telomere length, as expected, declines with age for both groups. And I will show you the box plots to convince you, hopefully, that these clocks are altered in people with HIV, even with viral suppression. We see that grim age, pheno age, Dunedin pace, these are all higher in our HIV group in red, controlling for demographic differences, and that telomere length predicted from DNA methylation is lower between groups. So these seem to be sensitive and useful biomarkers of biological aging in people with HIV. So what then does this tell us about the brain? I want to shout out my summer student, Isaac Kim, who did a really fantastic job with some of this work. He's from Johns Hopkins. So you'd be proud, Leah. The grim age we found in particular was associated with the brain age gap in such a way that having a higher grim age, in other words, a higher mortality risk as predicted from DNA methylation, is correlated with a older appearing brain age gap. And to look at this regionally, we quantified cortical thickness and we quantified regional volumes in a variety of cortical and subcortical regions from the MRI. And this heat map puts a more negative association in red. So hopefully you can see that the medial prefrontal cortex and the orbitofrontal cortex in particular, along with some temporal regions and some subcortical regions like the caudate, have a particularly strong relationship with grim age in this study. I'm gonna just double check the chat real quick. Interesting question about whether hunger would potentially affect this, since I'm guessing that that's a reference to the idea of fasting potentially being anti-aging. Ron points out that art doesn't fully normalize DNA methylation, and Michael's group has found that. I agree with that. I think I've seen that result repeated a couple times, that just getting on ART doesn't completely resolve epigenetic differences. And then Jules had another question asking whether aging and cognitive impairment is progressive for some people with HIV as they age. That's a good question. We don't have, even though we have some longitudinal data, we don't have a lot of extended follow-up. So the question of whether the biological aging differences are progressive is, to me, still kind of an unanswered one. Maybe this is just more because this is a cross-sectional data set and we're looking at different individuals over the lifespan. There might be more of a difference related to not to an individual's aging, but possibly to some cohort effects as well. Of, were you born in the in the 1960s and got HIV in the 1980s, and you spent a longer period of time without ART, maybe your aging process has been more affected, versus someone who might have been become positive in 2005, let's say, and maybe their brain appears to be a little bit younger because they got on ART right away, and so they had a healthier aging trajectory. It's something that I'm not sure about. Okay, lastly, does this actually relate to cognition? Because that's what this is all about. And we were interested to see that while most of the clocks didn't have a strong cognitive association, the grim age clock, once again, had a really significant correlation with executive function, which is one of the domains that we see taking a hit in older individuals with HIV, especially detectable HIV. We also saw that this is in fact related to social determinants of health. Once again, we have the area deprivation index on the x-axis, where a positive number is a higher deprivation, and we see that that is associated with greater grim age acceleration, and that education appears to have the reverse association, even more strongly than the brain age gap did. So we think that there's more going on than just HIV or viral load with DNA methylation, just as it is with the brain aging. So to summarize this part, we think that several epigenetic clocks are accelerated in virally suppressed people with HIV, consistent with some of the literature that is out there. What's really novel here, I hope, is the associations with the brain aging, which to my knowledge haven't been looked at in people with HIV. And this is opening up a lot of avenues to understand why people may be more resilient than others to aging while HIV positive and at risk for some of these other comorbid factors. So I know that we're getting low on time here. So just to give you a sense of where this could head next, we will definitely want to test what these two independent measures, epigenetics and neuroimaging, can contribute together to understand, to more accurately model and potentially predict brain health outcomes. especially at the level of individualized medicine. The question of whether epigenetic clocks can ever be clinically useful personalized biomarkers for the assessment of aging. And to ask what other underlying factors, I'm sure someone on here is about ready to ask this question, but what difference do various categories of substance use make for this epigenetic aging process? So I really want to thank first my mentor, Bo Anses, who's given me all these incredible opportunities to study these fascinating areas. And then my colleagues at WashU, at JHU, Missouri Institute of Mental Health, UC San Diego, the directors and former directors of the STAR program, and Tricia Berto at Rutgers. Um, I, of course, always want to appreciate the hundreds of volunteers who made these studies possible. And then finally, this is an interesting day to do the NIMH joint seminar series because this is day one of this government shutdown and we don't know how long this is going to last. So I want to give the most sincere thanks to the workers at NIH, in particular NIMH, who have been our partners in this process and have made this possible for us. And we are very much hoping that you guys will be well and return to work soon if you're furloughed. And I just want to thank everybody for taking the time to listen. I know we have some questions in the chat now.
Yeah, I was gonna say, Kaylin, do you want to, because you've kind of been answering throughout, so we can kind of wherever you stopped, and we can have people come on screen and ask their questions. This is a fabulous talk, and it's really amazing to see how far you've come with the biological clocks in relation to the brain age gap and everything else. So it's fun to see. Where did you stop in the questions? I think you answered Jules.
Yes. So I think I left off somewhere around Pilwa's question,
Okay.
which was just asking about, um, she says in the scope of epigenetic compaction, lamin could be another measure. I'll be honest, I don't know much about lamin, but I'm positive that there are other epigenetic assays that could measure things like chromatin accessibility, could measure histone acetylation, deacetylation. DNA methylation is just the easiest and most obvious thing to do at this level. But I would really love to one day collaborate with people who are doing things like ATAC-seq and other epigenetic assays. Um, Jules asks about IDU. I'm assuming that's in injection drug use history. Um, I'm not positive. Right now, we have an ongoing study, not of injection drug use, but of chronic cannabis use. But I think that history of injected drugs is something that we can't ignore. I know that we quantify the lifetime exposure of opiates, but I'm not sure whether the route of administration is something that we've ever studied. Um, let's see.
Ron
Ron
Ellis. Do you want to unmute? Because his face popped on the screen. So
Oh yeah. Hey,
make it
thanks,
interactive.
Caleb. That was very cool. Difference between people with HIV and without, not fully explained by the area deprivation index. You might have said it, but just wanted to check.
Yeah. Um, no. So
Okay.
it is one of the top ranking explanatory factors. It explains some of the variance in the brain age gap, but not all of it. So probably it is one of the mediators between HIV status, whether it's, I don't know if it's something we should conceptualize as being upstream or downstream, but or an independent third factor. But obviously our individuals with HIV have greater socioeconomic stressors than our individuals without HIV, even though they belong to roughly the same geographic community. So it is carrying some of the effect of that
Mhm.
HIV group difference, but not all of it.
Okay. And then I'm going to upstage Victoria. I hope you don't mind, Victoria, but I think her comment is very important. So this, I think it's called the Danish winter, when basically there was severe food insecurity that caused a generation to have a variety of epigenetic changes that then were passed on to their offspring who had obesity and diabetes and metabolic disorders. And, so passing on these things means now that women who have babies who have suffered under these conditions are going to carry the imprint of those effects. So very, very important, even if they have well-controlled HIV.
Yeah, to my knowledge, our data can't answer the question of
Yeah.
heritable differences in epigenetics. But I would imagine that particularly in famine-affected regions, that could be extremely important in modulating how the body responds to HIV. Whether you've had gene silencing because of food insufficiency that your parents or grandparents experienced. It's a really, really cool phenomenon to study, but I don't know much about the relationship between the heritable methylation changes and
Mhm.
HIV. Hey, Carrie. So Carrie
Yeah.
asked if we've explored sex-based differences in brain age gap. I know you have with the epigenetic clocks. Um, yeah, I saw that paper that came out earlier this year. Very, very interesting. We do see sex differences in the brain age gap. In general, our female participants have a healthier appearing brain age gap than our male participants. I don't know if this is linked to some sort of social difference, lifestyle difference, or if there's a simpler biological explanation of what's going on. But across the board, we tend to see female participants showing younger brain predicted ages.
Interesting. Yeah, we've just been looking at sex-based differences in some of our data from our cohort in New York, where we have pretty good representation of women. And interestingly found that women tend to have less epigenetic age acceleration. We
Uh-huh.
also look at the residual, which is kind of a scaled difference instead of the just absolute difference for
Yeah,
age,
fine.
chronologic and epigenetic age, which is I think another kind of helpful statistical approach. And I was curious about hormonal transitions across the lifespan in women, how that might interact with these amazing MRI data that you have and are really privileged to have. It's great.
Yeah,
It's
thanks.
exciting work.
Um, so we do, I believe, record menopausal status. And I know that there are several groups out there who are particularly interested in this question, including Pauline Mackey. I don't know. And I would be intrigued to find out whether there is a difference in the trajectories of computed brain aging when women start hormone replacement therapy, whether that affects in any way the morphology in such a way as to influence the model. It's just something that at this point I can't answer, but it's a really good question. Let's see. Carl asks about depressive disorder, which is a very good point. So I have looked to see whether our brain age gap biomarker is associated with depression. We have BDI in most or all of our participants. I didn't see any differences in BAG associated with higher depression. But we have looked at region by region in the brain, and we have found some regions that may be particularly sensitive to depression. But the idea of looking at it with the DNA methylation assays is very interesting. I I hadn't seen that result, that grim age is associated with depression, but that makes me really want to look at it. And Jules points out that the depression may be undiagnosed. So hopefully the research instruments would pick up on that continuum of depressive symptoms, even if people haven't gotten a formal diagnosis of MDD. Yeah, I'm sure I've missed some questions here, and I apologize for going through so much so fast. I hope I didn't leave everybody baffled and confused, but
No, this is fabulous. I just think you've got a lot of people, interesting carries on here. Nicole's on here. There, folks that are interested in the clocks, and you've got folks who are interested in the imaging. So I think this is a great discussion, and to see that what you're seeing with BAG and what Carrie's seeing, and that I guess, Carrie, that was the kind of the point I brought up to Caitlin earlier. Are there other measures? Because you're not just taking that difference, you're actually dividing it. So Caitlin, I'm wondering, if you apply that, does that change any of your pattern of findings to be more similar or different? Or is
Right.
there something very different, likely, Carrie, about your women, right, versus the folks in St. Louis, at
Yeah.
near Wash U? So that's why we need big databases for these questions.
Yes.
In our last few minutes, any other questions?
Yeah, if I didn't get to you, please raise your hand or shout it out.
If not, Kaylin, your presentation was absolutely fabulous. You've done a ton of work, and your K was just funded about a year ago. Just to see the momentum that you've had within the epigenetic space. So I can't wait to see, how things keep moving forward. And then Caroline, if you want to fill the group in on who's speaking next week, and I hope everybody comes back.
Is it okay if I ask a question quickly?
Sure. It's
Yeah.
Dr. Deb Gustafson from SUNY Downstate, and her talk is titled Cognitive Performance in Blood-Based Biomarkers in People with HIV. All right. Thank you, everybody. Thanks again, Kaylin.
Wait.
I think there was one more
Someone wanted
question.
to ask a question before.
Yeah.
Go ahead.
Yeah, so thank you very much for the talk. It's really, really interesting. So I am an imaging side. I'm also doing research in HIV with comorbidities. And what I saw in your talk, that you brought imaging and you brought DNA methods and sort of look into cellular levels. And so the question is that how does imaging in future direction for HIV research? You're already getting information from the cellular level. What role will imaging play in future research, HIV research, since you're already getting the most important part is the cellular level and the association with the cognition? So just that part out.
Yeah. I think I'd answer that question by invoking the idea of combining different imaging modalities. This idea that brain structure from T1 weighted MRI tells us only a limited thing. It provides us only a snapshot of a person's overall neurological health. But what if we were to combine that with functional MRI, potentially even with PET, to get a higher dimensionality of data about ongoing brain health processes, inflammation, for instance, or changes in functional network. I know that there are brain predicted age models that have been published that utilize fMRI as well as structural MRI. So I think that going forward, this is probably going to be a field where multimodal data becomes increasingly important, including in the MRI space.
Awesome. So thank you again for everybody for attending. This led to a lot of great discussion, and I look forward to seeing everybody next week.
Thanks, everybody.
Yes. Okay.